All right, it's going to be a little bit of a crash course compared to what we learned in at the beginning of this year. I am specifically looking at this requirement here. Sorry, it's not focused. There, can't see it anyway. All right. For those that are working on their internals, please be respectful to your classmates that are listening for their notes so that way they're prepared for the exams. All right, so I want to look at this bullet point here. So biological ideas, processes related to sources of variation within the gene pool selected from independent assortment, segregation, and crossing over during meiosis. So I need to potentially revise some of that material. First off, are you guys okay with meiosis versus mitosis, or do I need to do a quick rundown of the difference? Quick rundown? Yep. So, and that's fine because then it gives me stuff to tick off this list as well. So for mitosis, remember that happens in your toes. We have a cell, and that cell has DNA in it. It will double the amount of DNA it has. Try to make it more darker. After it then doubles that DNA, the cell will split into two. Now, with these two cells, do they have the same amount of DNA as the parent cell? So these are known as parent and daughter cells. So would this one have the same amount of DNA? We doubled the DNA in this round, and then in this round we half it. So when it splits into two cells, it will have the same amount of DNA as parents, or as parent cell. Will they be identical? Yes. These are known for having things like growth and repair in the body. And we call these body cells somatic cells. So are we okay with that so far? All right, looking at our checklist, I'm ticking off the word mitosis. I'm ticking off the word somatic. Yeah, we've done DNA. DNA is a double helix. All right, keep going. Yes. All right, meiosis is a little bit different. And that's the main one that we focus on. So meiosis, again, I have a parent cell with DNA. Something that it does as well is that it doubles that DNA. So things are very similar. Like that. Yeah? These guys will split and it will half the DNA. Like that. But, and this is known as meiosis 1, that first division. The other thing that's different is that these guys will split again. So we half the DNA twice. So So we have 4 cells. This is known as meiosis 2. So that's meiosis 1. This is meiosis 2. We have 2 cell divisions in this case. And we have 4 daughter cells. So we get 2 splits. Would these cells have the same amount of DNA as the parent? I've doubled, I halved, and I halved. So how much DNA would it have then?
A quarter.
But I doubled it first.
Yes, half.
So it's half. So these are my 4 daughter cells. They have half the DNA from the parent. Are they identical?
No.
No, these are non-identical. Which is good because then it means that we have more variety. And I can't spell identical right. What else do we have? This is for sexual reproduction. These are known as gametic cells. Or sex cells, which would be my sperm and my egg. All right, when does my DNA get restored to the right level? So if I have half in these cells, when does it get restored back to a full amount? When they double, do these cells start dividing and doubling and making the right amount, or do I need to have another parent?
You need another parent.
I need to have another parent. So this is half from the parent. So DNA amount is restored. During fertilization. So you get half from mom, you get half from dad, and together you get one full person, which is your offspring.
Yeah.
Yep. Gametic cells. All right. See what we can check off so far. Gametic. I should also add that these guys would be diploid. So it has a set number of DNA. Yeah, so they have the normal amount. Bring it back. So diploid, 2 sets of DNA, full set. Yeah, whereas this one would be called a haploid because you only have half the DNA.
Haploid for the other one. The $4.
Yeah, $4 are called haploids. I just want to tick off some more words. Haploid and diploid. I don't know why diploid's on that list twice.
Oh, 6 cells.
Should be an E. Alrighty. Can I move on? All right, for those that I'm letting you guys have study time, this isn't time to be on your phone. This isn't time to chit chat with your friends. This is time to get your work done. All right, move on. Okay. Shh. All right, the specific step that I want to talk about is meiosis 1, when we go from here to here and we half our DNA. Because there's two very important things that happen there that allow for more variation. Oh, it's trying to move my chair. All right, so when we had mitosis, and we were preparing for cell division, the chromosomes aligned in a single file line, right? Guys, please just be respectful for the other people in the class. They're trying to listen to me give notes. I'm trying to give you guys the tutorial time. All right, in meiosis, when they align, remember they're going to line up in homologous pairs first. So they line up as homologous pairs. Do you guys remember what homologous pairs are? When we see the word homologous, what does that kind of remind us? The homo. The same. So two of the same. Homologous pairs. Do you remember how many chromosomes we have? We have 46 total. How many pairs would that be? 23 pairs. We have 22 autosomes, and then one sex chromosome pairs. Does that ring a bell? It's going to zoom it out. So they align in the homologous pair, where in this case, all the chromosomes will just align in single file. Yep. Now, what we need to remember is that homologous pairs can align randomly. So, for example, I can have all the dark blue lining up on the same side, right? That's one random setup, correct? Or I could have it so that way they align. And I have, say for example, let's say I have this example. These two dark blue ones have aligned on the same side, but this dark blue one's on the other side. So you see how these are two possibilities? I'll just try to zoom it out so you can see the whole thing. You see how these are two possibilities for how they might align. So this is known as independent assortment. They assort independently from each other. And that means we can have lots of combinations of potential, blue and white ones being combined together. Okay? So this one here is showing independent assortment. So with that independent assortment, what we have is the homologous pairs line up on their own. So we get lots of different combinations when the cell divides. So what that means is that we get lots of different potential variation on combinations. And therefore that increases my variation. Just gonna freeze that so I can find a good picture of that for you. So I find my thingy. Too much of a mess on my desk. There we go, freeze. Or are you guys okay? I can move it. Move it? All right. Let me just show you guys an example of independent assortment. Just trying to find a good one. There we go. All right, so this is a little bit simpler because you have less to deal with. In this possibility, we've aligned the blue and the red together. Whereas in this possibility, we swapped around these two down here. And what it's doing is it's providing different potential combinations. That's independent assortment. Let me find you another picture that I like. This one should show it too. So this is the one that I was drawing. So you see how in this example it has all the blue ones lined up and all the red ones lined up. In this one it has swapped these two around, and this one stayed in the same spot as that one. In this case we've only swapped the top one around. Sorry, we swapped the bottom two around. So there's lots of different ways you can combine together the chromosomes. And remember, we have 23 pairs. So each one of those 23 pairs will independently sort itself. So that means you have 23 to the power of 2. So there's 529 different possible combinations that we can make from that. From that alone. But we all look different. All right, so that's independent assortment. The other thing that happens is crossing over. So when my chromosomes align during meiosis, 1, like that. So you remember how we had one blue one and one white one?
Yeah.
Yep. So an independent assortment, what happens is that some of this chromosome swaps for some of that chromosome, known as crossing over. So what happens is some of this will come off and get attached to this one, and some of this one will attach to that one. That's crossing over. So that's crossing over. And this is another source of variation. So what happens when genes are on a chromosome, they are known as linked. So say, for example, if I have a gene here, and that gene is for eye color, like big B, brown, and then I have another gene here for curly hair, let's say H. These two are known as linked genes because they're on the same chromosome. Do you think if they're linked, they'll be inherited together? Yeah, so if they're linked, they're inherited together. So often if you have like brown eyes, you might have curly hair. Yeah? So maybe, for example, on the white chromosome, I have the little b for blue eyes, and I have the little h for straight hair. Does that make sense? When I do stuff like crossing over, I'm then allowing the recombination of genes. So for example, in this guy here, we'll still have the big B, but now is going to inherit the little h in that combo. This one over here is still going to have the little b, but the bottom is now going to have the big H because it's come from this guy on the other side. So that is really good because it's another source of variation. It recombines linked genes. To create new patterns. Are you okay with that? All right, let me show you that. All right, so this is what we're looking at for crossing over. We have our homologous pair of chromosomes. Some of that DNA gets swapped, and now we get a new variety of combinations. That's really useful because when we start to make our new sex cells, just trying to find an example of one that I like. That one's probably too small. That's the one I want. There we go. So when we start making our sex cells, do you see how there's been some recombination and there's that mixture of red and green? And so all four of my haploid daughter cells, my sex cells, are different from each other because of that crossing over. And let me show you guys what I mean by linked and unlinked. It's just like I was showing you guys before. So we see how the A and the B genes are on the same chromosome, and you have the capital A with the lowercase b, capital A with lowercase b, lowercase a with uppercase B, lowercase a with capital B. When we have that crossing over and that recombination, I now can get the combination of having 2 capital letters and 2 lowercase letters. So that's why crossing over is important to allow for that variation. Are we good? Hopefully. All right, I think that was all I wanted to cover for today's lesson when we're looking at this. The other thing that we have to be mindful of is random fertilization. Remember that there's a random, the sperm and the egg just combine together randomly, so it's a random shuffle. You don't get specific, put this with that. So that's another source of variation, because if I'm going through the crossing over and the independent assortment and my partner's doing the same thing, then our genes are getting shuffled around, so that way our offspring will have a mixture of everything. It's a great way to create variety. All right, trying to think what else I wanted to show you. I think that's mostly it. The no brain too small, you guys can start doing those questions. I really like no brain too small because they collate the questions together, so they come into themes. So if we go to no brain too small and we go under genetic variation, you can see that they have collated questions. The ones that you guys can probably do are the genetic variation questions. So these are past exam questions that all look at variation as a source, variation in genetics. It primarily focuses on that meiosis and all that independent assortment, random fertilization, crossing over. So that's what these questions are all themed as. It's worthwhile at least having a read through all those questions so you can see how they ask the same question in different ways. But you guys should be able to answer these questions now. The other ones that you guys should be able to answer by the end of this lesson from the stuff that we did last week is the collated questions that are classified under other types of crosses. This is basically stuff that they weren't sure where to put, but all of the lethal alleles, multiple alleles, co-dominant, incomplete dominant, those questions are in here. So you can do these questions for the most part. I have noted which ones you can't do because we haven't quite done enough material. But this is basically everything that we've done in that first term. All right? So what questions do you have following up from that? Is there anything you guys need clarifying on? All right, on Google Classroom, I've put the pages that go to this lesson in particular. If you guys want to practice that stuff, you guys have about 10 minutes. So I would suggest getting a little bit of work done. I also want to check in with those that were working independently to make sure they got some stuff. And then we'll write in the list of the new vocabulary that we've covered. But the SciPad pages 104 to 106. And 108 to 113 is all looking at this material that I've covered in today's lesson. You could also, like I said, start working on some of those no brain too small questions if you want. And then just drop it into my box whenever you have printout works done, things like that. Cool? All right, that's it for us. This lesson is recorded, and I will scan all my notes in case you've missed something. Okay?